Hello student, welcome to EPG Pathsala. I am Dr. Ajit Jaswal from Department of Anthropology, Pondicherry University. Today we are going to discuss about a very important module, that is Introduction to Genetics under paper Physical and Biological Anthropology. Dear student, in this module we are going to develop and understand about what is genetics. We also have a brief idea about the structure and function of cell, and also have a comprehensive knowledge of DNA structure and its function. We also try to know the basic concept of chromosome and cell division. Let me give you a clear-cut information about genetics. The question that often came in our mind is why we are morphologically similar with our parents and different from member of other families. In the same way, there is another question, why member of the population group share some resemblance with each other and differ from those of other population group, or why such similarity and variation do exist among individuals. Is there is something that we inherit from our parents from the time of our birth, or do we share something with the member of our population group? The result in having similarities morphologically. Yes, we do inherit something from our parents that make us look alike with them. That sometime or something came to be known as gene. The word gene was first coined by Johnson in early 19th century. Without knowing the biochemical properties, but just because it is nice small word to describe something. And it was later on Bateson who defined heredity as similarity due to relatedness. The basic nature of the gene was described by an Australian monk called as the father of genetics, that is George Mendel. Gregor Mendel, more than a century ago, after a careful experiment conducted in a common sweet pea plant or called garden pea between 1856 and 1868, he summarized his finding in two laws. The law says that the gene was recognized as a particular factor. It is not something that passes unchanged from parents to the progeny. He described that each particular factor or gene exists in pairs in individual organisms. When an individual has two different particular factor or gene, only one of them expresses, and it is so said to be the dominant to the other, who is said to be recessive. During the gamete formation, the paired particulate factor separate or segregate randomly with equal likelihood. This is known as Mendel's law of segregation. The second law is known as Mendel's law of independent assortment. In this law, he described that during gametic formation, segregating pair of a particulate factor is short independently of each other. That is, each paired gene freely separated and do not influence the separation of other paired gene during gamete formation. These two laws explain the basic fundamental principle of inheritance. However, his groundbreaking research was discovered only in 1900 by two independent scientists. One is William Bateson and Hugo de Varis. 16 years after the death of Mendel and 35 years after he published it. After the discovery of Mendel's idea of inheritance in 1900, a whole new science called genetics, and Bateson coined the term in 1908, emerged. So the genetics is that branch of science which deals with the study of inheritance of unit factor are called gene. These genes are present inside the nucleus in the form of DNA in every cell. Cell. Cell is the basic unit of every life. It is a microscopic organism, organic body which carries genetic material and other organelles required for cellular function. All living organisms are made up of cell. Some are made up of single cell and called as unicellular organisms, like bacteria and protozoans. This single cell performed all the activities of being alive. Other, including human, are made up of multiple cells and hence called as multicellular organisms. Among these organisms, different activities are performed by different specialized cells. All the living organisms on earth can be divided into prokaryotes and eukaryotes. Prokaryotes are those organisms with no major compartment within the cell to separate the genetic material from the other component of the cell. Bacteria, blue-green algae are some of the examples of prokaryotes. Prokaryotes are the earliest form of life on the earth, appear in the fossil record of about 3.4 billion years ago. On the other hand, eukaryotes are characterized by having nucleus that separate the genetic material from the rest of the cell component. In the fossil record, eukaryotic cells do appear in about 1.5 billion years ago. Eukaryotic cells resemble to those found today. Evolved somewhere around 850 million years ago, and multicellular organisms made their first appearance only 600 million years ago. In multicellular organisms like human, different specialized cells perform different functions. For example, brain cell or neuron is structurally different from blood cell based on their different function. However, cells in a complex multicellular organism can be divided into two types, that is somatic cell and a gametic cell. Somatic cells are body cells and do not directly involve in reproduction. Somatic cell again can be of different types based on different function, whereas gametes are the sex cell which directly involved in reproduction. In human there are two types of gametes, one is egg, another is sperm. The egg is called as female gamete cell and the sperm is called as male gamete cell. Cell anatomy. Generally, eukaryotic cells share a similar structure. Every eukaryotic cell has a semi-permeable membrane called plasma membrane that bound all the components of a cell together. Inside the cell, all the organelles are float on a gel-like substances called the cytoplasm. The most important and prominent structure in the cytoplasm is the nucleus, which has its own thin envelope called nuclear membrane, separating its content from the cytoplasm. Inside the nucleus contain two complex molecules called as DNA and RNA. DNA are called as deoxyribonucleic acid is a double-stranded complex molecules, where genetic information is stored, whereas RNA is a ribonucleic acid is similar with DNA but have single strand. RNA is also found in cytoplasm. It is essential because it becomes the template for protein synthesis. Another important organelle found in the cytoplasm is mitochondria. The main function of the mitochondria is energy production, and hence is known as powerhouse of the cell. It is a capsule-shaped structure and carry its own genetic material called mitochondrial DNA or mtDNA. Structurally and functionally, mitochondrial DNA is similar to the nuclear DNA, but differ in the pattern of organization. And mitochondrial DNA is an important tool for evolutionary studies. It follows the mate RNA one line of inheritance, that is inheritance from mother to her progeny only. This is because father's mitochondrial DNA does not involve in zygote formation. Another complex structure looks like a folded sheet called endoplasmic reticulum or ER is found in the cytoplasm. The main function of endoplasmic reticulum is to increase surface area within the cell for metabolic reaction to take place. Ribosome is another organelle within the cytoplasm which main function is protein synthesis. Ribosomes which are attached to the endoplasmic reticulum is called as attached ribosomes, and those found floating in the cytoplasm as free ribosome. This figure shows the different component of the cell like the plasma membrane that bound all the cell component together, the cytoplasm, a gel-like structure that fill the cell. All the organelles float in the cytoplasm. Nucleus, which carry the genetic material, it has a thin envelope that separate nucleus from the cytoplasm. Mitochondria, which is known as the powerhouse of the cell, located in the cytoplasm. Metabolic activities are carried out in it. It has its own DNA called mitochondrial DNA. Endoplasmic reticulum, it is found in cytoplasm, gives extra surface area for metabolic reaction. Ribosomes are actively involved in protein synthesis. It is present in the cytoplasm. Structure of DNA. DNA is the hereditary material contained inside the nucleus. DNA carries the inherited from parent genetic information required for life. DNA directs all the cellular function of an organism through protein. So, in order to understand the mechanism of genetics, it is necessary to know the structure and function of DNA molecules. The exact physical and chemical properties of DNA molecules were successfully discovered by two young scientists, James Watson and Crick, in 1953. Watson and Crick developed their model of DNA based on some of the properties of DNA which has already established by other scientists. In the late 1940s and early 1950s, biochemist Chargaff and his colleagues have discovered that DNA consists of 4 different nitrogenous bases, and these bases are distributed in a unique pattern. In a given sample of DNA, the amount of adenine is similar to the amount of thymine and the amount of cytosine to the amount of guanine. Another scientist, Rosalind Franklin and Wilkins, were also successfully able to produce an X-ray crystallography of DNA molecule clearly showing that the structure is helical. Discovery of DNA molecular structure have altered our understanding of biological and evolutionary mechanisms and completely revolutionized the field of biology and medicine. For their famous discovery, Watson and Crick and Wilkins were awarded Nobel Prize in 1962, but Rosalind Franklin dies 4 years before the award at the age of 37 years because of cancer. The structure of DNA molecules is double strands forming a double helix resembling a ladder twisting around its central axis. Each strand is made up of a chain of nucleotides. Nucleotide is the basic unit of DNA and RNA molecules. Each nucleotide is again made up of three components, that is a sugar, that is in the case of DNA it is deoxyribose, a phosphate unit and a nitrogenous bases. There are four different types of nitrogenous bases belongs to two classes, purines and pyrimidines. The purines are adenine and guanine and the pyrimidines are cytosine and thymine. Based on these four different nitrogenous bases, there are four nucleotides which can be represented by the initial letter of each base, that is capital A, capital G, capital C and capital T. In a nucleotide, each phosphate is bound to a sugar molecules and each sugar molecules bond with a nitrogenous bases. The nitrogenous base of a strand form hydrogen bond with the nitrogenous base of complementary strand, thereby resulting double-stranded DNA molecular structure. The bonding between the nitrogen bases followed a fixed pattern and it is always A to T and C to G, as it is possible to form two hydrogen bond between A and T and three hydrogen bond between C and G. In other words, the combination is between purine and pyrimidine. Purine are larger molecule than the pyrimidine and therefore this combination is necessary to maintain a constant distance between the two strands. For example, if the strand of DNA has a sequence of nucleotide A, T, T, C, G, C, A, G, C, the corresponding strand will have T, A, G, C, G, T, T, C, G sequence of nucleotide. So, if we consider the analogy of a ladder, the side of the ladder is represented by the chain of sugar and phosphate. Rungs are represented by the nitrogenous bases and bonds forming between them. RNA is similar to DNA except that first, it is a single strand unlike DNA which is a double strand. Second, deoxyribose is replaced by a ribose as a sugar in nucleotide. And third, pyrimidine base thymine is replaced by another pyrimidine base uracil and bonds with adenine. This figure shows the structure of DNA molecules. As the genetic material inside the nucleus are DNA and RNA, the chemical and physical properties of DNA molecules was discovered by Watson and Crick in 1953. The structure of DNA molecules is double helix, resembles a ladder twisting around the central axis. DNA is made up of a double strand. Each strand is made by a chain of nucleotide. Nucleotide is made up of a three component, sugar, phosphate unit and nitrogenous bases. There are four nitrogenous bases, that is adenine, guanine, cytosine and thymine. Adenine and guanine belongs to purine class, guanine and cytosine and thymine belongs to pyrimidine class. Okay, now let's discuss about the function of DNA. DNA, which is a genetic material. The genetic information to build up an organism is transmitted and stored as a sequence of nucleotide called DNA or deoxyribonucleic acid from the parent to progeny. So that it is the DNA which carry the information from the parents to the progeny. This complete set of genetic information for any organism is called as genome. So genome is nothing, it is just a set of genetic information. Genome itself does not perform any active role in the functioning of the organism. Rather it carries the information for producing protein in the appropriate times and date. So this set of genetic information act as a carrier for the production of protein. So all the cell of an organism should carry the exact genetic information. In order to achieve this, DNA should be able to replicate itself and also should be able to decode the information for protein synthesis. So overall we can say that DNA is nothing, it is a genetic information carrier, and which undergo a process of replications, later on transcription and translation, ultimately lead to the formation of protein, which is the ultimate product or the coded information of the genome or DNA. DNA replication. Every multicellular organism starts from a single cell zygote. This single cell zygote was divided billion times to form a complex organism. So when a mother cell divided into two daughter cells, an exact copy of genetic material such as must be transmitted to each daughter cell. The structure of DNA molecules suggests the mechanism for its replication. Replication of DNA starts with the separation of double strand into two separate strands by breaking the weak hydrogen bond between the nucleotide. Each of these separate strands act as a template for the formation of new complementary strand of DNA by assembling nucleotides. Every step of DNA replication is mediated by a particular enzyme. After completion, two copies of mother DNA molecules are formed, each having one original strand and one newly formed strand. These two DNA molecules are then passed on to two daughter cells during cell division. Protein synthesis. The cellular function of an organism is executed by various proteins. For example, hemoglobin found in a red blood cell binds the oxygen molecules and carried to cell throughout the body. Similarly, insulin helps in the metabolism of sugar molecules in the body. The production of these proteins in different time and place is possible only by following the genetic information stored in the DNA through the process called protein synthesis. Proteins are complex molecules made up of a smaller molecules called amino acid. Amino acids have the ability to form long chain by bonding to each other. There are 20 amino acids, of which 9 are not able to synthesize in the body and have to be obtained from diet. These are known as essential amino acids. Remaining 11 non-essential amino acids are produced inside the body. These amino acids line up in a particular sequence to produce a specific protein. By now know that nucleotide sequence of DNA carry the code for protein synthesis, and protein is made up of a chain of amino acids. So the sequence of amino acid is ultimately determined by the sequence of nucleotides. In DNA, a triplets of nucleotides or group of 3 nucleotides specifies an amino acid. For example, if the sequence of nucleotide is CGA, then it specifies the amino acid alanine. Similarly, a sequence of TTA, GGA, CTT, GTC, CTA, AGA will give a chain of amino acid, that is asparagine, proline, glutamic acid, glutamine, aspartic acid, serine. A typical protein may be formed by a chain of about 200 amino acids. The process of protein synthesis involves two steps: transcription and translation. Transcription takes place inside the nucleus. In this step, the nucleotide sequence coded for the particular protein is copied in the form of RNA called messenger RNA or mRNA. This mRNA exactly copies the nucleotide sequence of the DNA and becomes the template for assembling the corresponding amino acid. The second step, translation, takes place in the cytoplasm. So the mRNA come out of the nucleus through the nuclear membrane into the cytoplasm. In the cytoplasm, mRNA is bound at the ribosome and its information is translated in group of 3 mRNA nucleotides called codons. Each mitochondrial DNA codon specifies one amino acid like a triple do in DNA. Another RNA called tRNA or transfer RNA help in forming amino acid chain in the ribosome. Each tRNA molecules bind to one specific amino acid. These tRNA molecules take the amino acid that matches the codon being translated to the ribosome. These amino acids join together in the sequence dictated by the sequence of mRNA codons. In this way, amino acids form the required chain to be a protein. The given figure explains the process of DNA replication and protein synthesis. Replication starts with the breakdown of double strand into a single strand, achieved by breaking the weak hydrogen bond between complementary nucleotides. Each separate strand acts as a template for forming new complementary nucleotide sequence. After completion, 2 new copies of mother DNA resulted, each having one original strand and one newly formed strand. Every step of replication is mediated by a particular enzyme. Proteins are complex molecules formed by smaller molecules called amino acid. There are 20 amino acids. Each amino acid is specified by a triple of nucleotide in the DNA cell called codon. For example, protein synthesis takes place in 2 steps, that is transcription and translation, started inside the nucleus and ended in the cytoplasm. Transcription, it takes place inside the nucleus. The nucleotide sequence in the DNA code for protein is copied in the form of mRNA. Translation, in the cytoplasm, mRNA attached to the ribosome and codons are translated into amino acid. tRNA bring amino acid from that matches the codon being translated to the ribosome. Amino acid form required chain in the sequence detected by the sequence of mRNA codon to become protein. Chromosome. DNA inside the nucleus existed a dispersed and uncoiled strand called chromatin. During cell division, this chromatin becomes condensed, coiled and tightly wound into a discrete structure called chromosome. Each chromosome has distinct shape and size, which are partially determined by the position of centromere, a condensed and constricted region on the chromosome. The number of chromosomes is fixed for every species, so every human has 46 or 23 pair of chromosomes. Each somatic cell has 2 copies of each chromosome or diploid number of chromosomes, inherited one each from the mother and father. But gamete cell has haploid number of chromosomes, that is half the number of chromosomes. The gene or the fragment of DNA coded for a protein is distributed across the chromosome. The location of a specific gene on a chromosome is called locus. Since somatic cell has 2 copies of chromosome, each gene is also exists in 2 copies, one at each copy of the chromosome or locus. The alternative variant of a same gene is called allele. The given figure shows structure of chromosome and DNA. DNA inside the nucleus existed as dispersed and uncoiled strand called chromatin. Chromatin condensed, coiled and tightly wound into a discrete structure called chromosome during cell division. Number of chromosomes is fixed for a species. There are two types of cell division. First is mitosis and second is meiosis. Let's first discuss about the mitosis. Mitosis cell division occur in somatic cell when it replicated to form two daughter cell. It required for growth of the organism. Healing of wounds are whenever new cells are required to replace the old one. The life cycle of a cell can be divided into different stages. When the cell is not involved in cell division, it is called interphase. The first stage of mitosis is called the prophase, characterized by disappearance of nuclear membrane, condense of chromatin to form chromosome composed of two identical sister chromatids. This is followed by metaphase, where the chromosomes are aligned at the center of the cell. Then the sister chromatids are split apart and migrated to opposite end of the cell in anaphase. The final stage is called telophase, where the cytoplasm split, forming two completely separate daughter cells. Not every somatic cell undergoes mitosis. Red blood cell, brain and nerve cell, that is neuron, and liver cell usually do not reproduce once it is fully developed. Red cells are continuously produced inside the bone marrow. Meiosis. Meiosis has two divisions forming four daughter cells with haploid number of chromosomes, unlike forming two daughter cells with diploid number of chromosomes in mitosis. It occurs in the specialized diploid cell in testes of male and ovaries of female. This haploid gamete produced through meiosis is necessary because when a sperm and egg fertilized to form zygote, the original diploid number of chromosomes is resolved. The first meiotic prophase is similar to the prophase of mitosis, but with some critical differences, like pairing of partner chromosome forming four chromatids or tetrads. Pairing of chromosome is an important phenomenon because exchange of genetic information called crossing over takes place. After crossing over, cell begin to divide. The member of paired chromosomes separate and migrated to opposite end of cell, forming two daughter cell, but not identical to each other or to mother cell. Each cell contains only a member of paired chromosome or haploid. The second meiotic division executes in the same way as mitosis occurred, resulting in four daughter haploid cells, two from each two daughter cell of the first meiotic division. Okay, now let's try to revise, recap or summarize some of the important points related to genetics. Like, the word gene was first coined by Johnson in early 1900. Without knowing the biochemical properties, he called it as a something. But just because it is a nice small piece of word, he described this as that something which carries the information. After the discovery of Mendel's idea of inheritance in 1900, a whole new science called genetic emerged. Genetic is the branch of science which deals with the study of inheritance of unit factor called gene, which earlier called as something. Cell is the basic unit of every cell, every life. Cell is actually a complex multicellular organism that can be divided into two types: somatic cell or body cell and germinal cell or gametic cell. Somatic cell are body cell and do not directly involve in reproduction. Somatic cell again can be of different types based on different function, whereas gametic cell or gametes are the sex cell which directly involve in reproduction. DNA, which is a hereditary material, contained inside the nucleus. The exact physical and chemical properties of DNA molecules were successfully discovered by two young scientists called James Watson and Francis Crick in 1953. The genetic information to build up an organism is transmitted and stored as sequence of nucleotide called DNA from parents to the progeny. This complete set of genetic information for any organism is called as genome. There are two types of cell division, mitosis and meiosis. The mitotic or mitosis cell division occur in the somatic cell where it replicates to form two daughter cells. It is required for growth of the organism. It is also required for healing of wound or whenever new cells are required to replace the old one. Next type of division is called meiosis. Meiosis has a two division phase forming four daughter cells with haploid number of chromosome, that is n number of chromosome, not diploid in the earlier case. Unlike forming two daughter cell with diploid number of chromosome in the case of mitosis, it occurs in the specialized diploid cell in the testes of male and ovaries of female. So, genetics is nothing, it is a specialized or developed or advanced branch of science which mainly concentrate on explaining about the concept of inheritance, how the characteristics or the feature transfer from offspring, a parent to offspring, why the children look alike like their parents, what is the reason behind this. There are many concepts, many theories, many models which has a great confusion regarding these things unless and until the explanation given by Mendel's. Because of that, Mendel is also known as father of genetics. Mendel clearly explained in his theory that there is a transmission of inheritance of character takes place from the parents to the offspring or to the children. Thank you.